Oxalates make or break biofilms. Found in high concentrations in foods like potatoes, these
So here's another video on biofilms, and in this one I'm going to be talking mostly about pecans. So basically, pecans, they do have an effect on biofilms. So a recap on what biofilms are: they are a protective layer on pathogens and cancer cells that protects them from immune cell attack, such as macrophages, phagocytosis, which is like an engulfing attack on the cancer cell. So that the cancer cells are driven to areas of high inflammation. High inflammation is actually known to promote the formation of biofilms, and that is how the cancer cells are protected by these biofilms. Now, this is where pecans play a rather critical role when it comes to dissolving biofilms as compared to other foods. Other foods, they can reduce biofilms, but adding pecans in with those other foods, which I've talked about in my other videos, but adding pecans in can have a much bigger positive benefit and impact. That's because pecans contain copper, and copper is critical for iron metabolism within the lysosomal membranes. So this is especially critical because iron cannot get trapped in macrophages, and that'll make a whole biofilm protecting cancer cell scenario or situation a lot more worse, because if the iron is not being metabolized by copper, then the iron does get trapped, therefore leading to iron deficiency and then eventually hypoxia. And then the hypoxia drives the macrophages towards an M2 phenotype, which actually protects and supports tumors. So that would be a even worse situation if the macrophages are protecting the tumors while the biofilms are also protecting the cancer cells at the same time. Also, in order for iron to be exported out of macrophages to stop the iron deficiency, that an exporter called ferroportin must work. But something called hepcidin degrades that exporter called ferroportin. Therefore, the iron stays trapped in the macrophages, leading to iron deficiency. That's why it's important for copper to function correctly, so that the iron can be properly metabolized for the ferroportin exporters to also function correctly. Okay, so the way biofilms form is with bacterial adhesion, and but polyphenols in pecans, they may interfere with this bacterial adhesion. So the polyphenols in pecans, they bind to bacterial surface proteins, or EPS. EPS is short for extracellular polymeric substances. Therefore, the polyphenols binding to these EPSs reduces the ability of bacteria to stick to surfaces. So there's another thing called quorum sensing that actually is one of the signaling pathways that regulates adhesion molecule expression, or in other words, promoting biofilm formation. But the polyphenols and pecans are known to degrade that quorum sensing signaling pathway, therefore reducing the biofilms. So it's important for the biofilms to be reduced, because biofilms contain bacteria that actually sequester and rob nutrients away from the body. But one of those nutrients that is especially impacted and hoarded by biofilm bacteria happens to be iron. And with iron deficiency can eventually lead to serious issues because of vitamin B12 deficiency, because iron is involved in the sequential pathway that leads to adequate vitamin B12. And of course, if biofilms and the bacteria are hoarding the iron, and as well as macrophages hoarding iron as well, if the ferroportin exporter is degraded by hepcidin, then that's going to lead to iron deficiency, because vitamin A requires iron, so that vitamin A can make the parietal cells secrete intrinsic factor, because vitamin B12 cannot work without intrinsic factor. So vitamin B12 deficiency could lead to advanced glycation end products forming, because vitamin B12 deficiency leads to a glutathione deficiency, and glutathione is required for reducing advanced glycation end products. Because if those are too high, then the ECM, short for extracellular matrix, will become too stiff and rigid, and then the immune cells are going to be excluded outside of the tumor, because high ages because of B12 deficiency creates a stronger or more rigid cross-linking in the ECM. That excessive cross-linking will exclude immune cells outside of the tumor, and then the T cells cannot invade or migrate through that thick, rigid cross-linking. So another problem with biofilms is that if it's sequestering vitamins, because the biofilms could cover the mucosal layer where the nutrient diffusion normally happens in the gut, then vitamin C will also be impaired. If vitamin C is impaired, then iron is also impaired as well, because vitamin C is responsible for keeping iron in a bioavailable form. That way, vitamin A can function correctly. And another issue with the biofilms is that it even prevents the absorption of amino acids. And amino acids are required for protein synthesis for T cells, because T cells, they require amino acids for protein synthesis, also for like a tryptophan, to avoid tryptophan deficiency, because T cells, they can be depleted by tryptophan, which can lead to deactivated T cells. So this situation can be even worse if the iron is also trapped in the macrophages. And another issue with the biofilms sequestering vitamins is that it could be also sequestering zinc, which is going to lead to a thymus deficiency, or the thymus isn't really producing the T cells, because the T cells train at the thymus. The thymus is like the training camp of T cells. But of course, that's not possible if zinc is being sequestered by bacteria within the biofilms. So another great way to aid Pecans with the copper, to improve the copper absorption and function for iron metabolism, is to reduce this inflammatory cytokine called IL-6. By reducing IL-6, that also reduces the hepcidin that is degrading the ferroportin. That way, the ferroportin is intact, so that the ferroportin can export the iron that are trapped in the macrophages. A good way to reduce IL-6 is with a variety of different fruits. Fruits, they can help reduce IL-6, therefore reducing hepcidin. Because if the iron is not exported out of the macrophages, then the lysosomal membrane will not function correctly, because it does need iron to function. If there's no iron, then the cathepsins, they leak out of the lysosome, and then the exosomes cannot be degraded inside the lysosomes, because the cathepsins are responsible for degrading exosomes. Now, if the exosomes are not degraded, the tumors can receive, and cancer cells, they get the exosomes, and exosomes are also involved in biofilm formation, which is another rabbit hole for a different video for a different day. So that's it for the video. If you're interested in more future videos, then like, comment, share, and subscribe, and keep it locked in here on my channel, and I'll see you in the next video.